Hello, good morning, everyone. Welcome to our new webinar at Netri. We're going to talk about CNS and PNS application. So let's start. So hi again. I hope you see well the screen. That should be the case. So today we're gonna discuss about the latest information that we can share with you about the applications on CNS and PNS, and we have here to talk about it our expert fellows from the company. So we have Benoît Maisonneuve on my right, we have Helene on my left, and we have Jessica also on my right. So they will basically present you the different topics, and you can see here the CVs and all the experience that our fellows have about the topics we're going to talk about. So the program today will be first to discuss about the CNS applications, and we're going to scrutinize a bit more about the modeling of the Alzheimer disease and also neuro-immunological communications. Afterwards, we will have another session that will talk more specifically about the applications in PNS and more particularly the traumatic injury and CIPN-on-MEA electrophysiology. And of course, we will have a session of question and answers. So basically like 20 minutes of each session. So that will give you a lot of time to interact with us. Just to refresh a bit the technology that we're going to talk about and the platform that we developed on this technology. So Netri has developed and designed since the last 6 years a technology for high throughput and also interoperable solutions. So the device that you are seeing here is composed of up to 16 chips, which are identical. And those chips, we will talk about a bit later in the next slide about the technology. The chips allow for each chip to have experimental condition. So that means that you can run up to 16 conditions in one running on one device. The main advantage and the message to take home about our technology is that it is a very accessible technology in terms of easy to use, as we use a specific material which is PDMS, and we have know-how to manufacture it. And that means that because of the permeability of the gas from the PDMS, we have the exchange, for example, of oxygen, and so the cells can breathe. So there's no need to pumps and standalone devices to make the cell culture run. The second thing that we have in our technology is the capability to compartmentalize the culture between each other. So that means that when you do a co-culture or tree culture, each culture can be run separately into the same chip, and then we can look at some interactions that we will talk in the next slide. And finally, the format that is used for our technology is a 96-well plate format. That means that the compatibility with the lab equipment is full, whatever from robot handling, if it's liquid handling, imaging, and so on. So the idea behind the technology of Netri is to be capable to use also the neurons as a sensor, as a biodigital sensor. That means that the organ can be addressed using this high potential technology. And so we'll have, in addition to that, also dynamic responses and also non-destructive, specifically if we talk about the electrophysiology. So finally, we are building a digital library that will allow us ultimately to be able to provide a model of productivity and efficacy as well as toxicity. So just to finish on that part, the technology itself is based on, as I said, one chip composed of 6 wells, And so each chip is composed of 3 channels that are, as I said, physically separated and independent. So the compartmentalization is achieved. And in addition to that, we have created micro channels that allows at the interchannel, so between the channel 1 and the channel 2, and between the channel 2 and the channel 3, that allows the axons of the projection of the neurons to progress from their channel of seeding towards the channel where they can innovate. So that's really the idea behind. So you would see also that in other previous webinars that we have the capability to use also 3D architectures. But at this stage, what we want to focus on is the capability of using high throughput and also the possibility to use electrophysiology in our devices to measure the electrical activity of the neurons within this region of interest. Of course, if you need more details about the technology, we will be more than happy to answer to that either at the end of the seminar, the webinar, in the session of questions and answers, or separately after the webinar. So as a summary, our capabilities and our global offering is based on the technology that we have provided through the organ-on-chip or organoid-on-chip that we are capable to produce. We can allow you to use our product directly in your labs using either the microfluidic potential that we have, and additionally, as an option, the electrophysiology, which is implementable in our devices. And additionally to that, we also provide large services for screening and co-development in all labs at Netri, enabling you to develop a platform or do drug screening. So you see the applications are quite vast, and the potential is also very important. So we're going to just focus on a few of those applications today. So after recapping very quickly the technology at Netri, then I would like to give the word to Benoît that will present the CNS applications and more specifically on the AD modeling and neuro-immunological communications.
Thank you, Thierry. And once again, hi, everybody. Thank you for joining us. So we're going to go through quickly 2 of our CNS applications, as Thierry mentioned. Without further ado, let's jump straight into the Alzheimer's disease application. So I'm not going to go in depth into what Alzheimer's disease is, because I'm fairly certain that most of you actually have a pretty good idea of what it is and the state of knowledge that is that researchers have at the moment. It's a CNS condition, and the thing to keep in mind is, for now, what is studied most are amyloid beta accumulation and anti-nasopathy as well for most of the patients. These oligomers that are predominant, that can be neurotoxic in brains during early stage of AD, and focusing on that enables us to develop a model, and then that can be used in order to help drug developers to find suitable strategies in order to target this and hopefully hinder the progress of the disease. So we use actually a couple of different devices to start to work on the model. The first one I'm going to talk about is the one that we call the neuro-immuno. So that's the one you can see on your screen. So it's a 3-channel device that has a MEA interface that enables us to record the electrical communication between the cell populations. So what we did for this first part of the work was to use glutamatergic neurons and GABAergic neurons. So we use neurons from brain cells, and we adapted the cell culture protocol to our devices. So first independently, and then in co-culture. So what you can see on your screen is what we had from the adaptation from the supplier protocol, and you can see that the cells look fairly happy and they grew really well. When we put them into core cultures, well, they kept on actually feeling pretty good. So what you can see here, for example, on the left-hand side of the device are glutamatergic neurons, and on the right-hand side are GABAergic neurons. And what you can see as well on the right-hand side on the graph is the expression of very specific markers at day 21 that confirmed us that the differentiation process went well and that we have the population that we are targeting in each channel. So you can see some mainly but with the VGLUT one that is highly expressed in the channel 1, which is the one on the left-hand side, and the GABA that is quite highly expressed in the channel 3, which is the one on the right-hand side. So having this core culture is good, they look good, so the physiology is nice, but what is important for us is to make sure that we are able to record what's happening inside of them and how they communicate. So here we can see actually evolution of recordings that we had throughout the culture to make sure that they reach a point when they are mature enough to actually have an electrophysiological activity. What you can see here is as we culture them, so from day 7 all the way to day 21, an increase in firing rate in the neural network. The recording the activity actually starts to be fairly good from day 21, but we usually push it all the way to day 14, but we usually push it all the way to day 21 to make sure that we have maximum activity. The good thing that we could see from this kind of exploration was that there was no cell damage that was due to repeated recordings. So that's a really nice thing because, as Thierry mentioned, the whole idea of using MEA as your readout is that it is non-destructive, so you can actually keep on going with your experiment and hopefully be able to actually see what's happening on longer time frame than just a single point when you are doing a fixation or anything. So as I said before, the differentiation process went really well. So we had over 70% of the phenotypic markers that we were expecting, and we had less than 10% of the potency markers when we were comparing the staining with early seeding. So having that gives us actually a robust base on which we can work on in order to be able to start to develop platforms that will emulate some of the tauopathy or alpha-synuclein accumulations. So that's something we are actually doing in collaboration with ETAP Lab within a project that is called BioDiamond. So a lot of the work linked to the electrophysical activity is still ongoing, and and we are not ready yet to explain to you, to show you a bit more what's happening, but that should be coming soon. In the meantime, we also explore different ways of using this kind of architecture and this kind of culture in order to start to explore the effect of habitat oligomer addition to the culture. So the work I'm going to show you in the next few slides has been carried out by our partner, as I mentioned before, ETAP Lab. And they have been using another device called the Dual-Link Shift. This one is slightly different in the way that the center channel is not exactly in the middle of the two other channels. It's slightly shifted on one side. That enables us to actually have what we call a synaptic compartment and be able to focus more on this. So here you can see, for example, a rodent culture, a neuron rodent culture that has been either just in culture by itself or culture with some beta peptides that they're all producing that is produced by ETAP Lab. And there are two main things that are interesting here. The first one is that we don't see any cellular death in the axonal in the soma compartments. And on the axonal, in the axonal compartment, so the one on the left-hand side, we can see degeneration. So that's in a dose-dependent way. So that's a good way for us to make sure that what we will be able to see later on in the project is not going to be linked to cellular death in the soma compartment, and it's really linked to what's happening in the axonal compartment. So there's a few next steps that are coming up. Of course, as I mentioned, so there's a lot of work that is still happening on the Alzheimer disease one, especially using actually their peptide that they are producing. And hopefully we'll be able to share more information on that soon. But there are also other pathologies that we are exploring within this collaboration. There's ALS, and we are currently working on a platform that enables us to co-culture motor neurons and muscle cells in order to recreate an NMJ platform. But there's also Parkinson's disease that we're interested in, where we will co-culture dopaminergic neurons and GABAergic neurons. So I hope I will be able to share more information on that with you very, very soon. In the meantime, we'll stay tuned. There will be more information and actually further work for those of you that will be present at SFN early next month that we should be able to show you on this specific subject. Now let's jump actually into something else, the neuro-immunological communication part. So the work I'm going to show you now has been done in collaboration with the University Hospital of Saint-Étienne in France. So everything I'm going to show you has been published. You will have the reference of the paper if you want to go more in depth in the coming slides. So the overall idea behind it for them was to start to go towards inflammatory bowel disease models, or at least platform, or things that will be able to enable them to actually explore the impact of chronic inflammation on various organs, of course, the digestive system in the case of IBD, but also on, globally speaking, on neuronal communication. So there's a lot of intricate way that this can be addressed, and there's a lot of different parameters that needs to be explored. The one that the researchers have focused on during this work really was inflammatory suppression, immune cell activation, so more specifically dendritic cells, as we will see later, and this impact on neural communication. So as you will be able to see, so here you have actually the name of the first author, and you should be able, using the DOI, to find the publication really easily. What they've done was they did a co-culture of glutamatergic neurons. So they seeded glutamatergic neuron in channel 1 and the channel 3, so the one on the left-hand side and the one on the right-hand side. And then they produced to carry out several experimental plans, so by adding LPS, for example, in some channels, but also by adding some immune cells, more specifically the dendritic cells. And then they looked at how the cells behave, but most importantly at how they communicated between them. So first, a quick look at the cells. So they developed fairly well, as you can see in the devices, which was to be expected, actually, if you remember the slides from a few minutes ago that I showed you. And on the right-hand side, you can see that when they added dendritic cells, everything went well. They really stayed in one channel as expected. So the compartmentalization really does its job here. And they actually look really, really nice. So what they did then, they wanted to make sure that they were able to see if there was inflammation that was related. So especially if the dendritic cells that were activated before being seeded with the neurons were able to secrete some inflammatory compounds. And so the way they checked that was by doing immunoassay on interleukin 6. And what you can see here is you only detect interleukin 6 in the channel where the dendritic cells are added. So that really showed that first we have it, but it's really, really specifically in this channel. So once again, the compartmentalization is doing its job, and you can really start to explore actually what's happening in each channel independently. I'm just going to have a quick few words on the graph at the bottom. So that's actually part of the one of the interesting parts of this proof of concept study. That was to try to see if we were able, using our devices and MEA, to see how dendritic cells or immune cells could actually influence electrophysiological activity. And what they've seen is that there was an impact actually of adding dendritic cells on glutamatergic neurons. Once again, keep in mind that the dendritic cells were activated for this experiment. And what you could see is condition 2, for example, was the condition where dendritic cells were added into the channel 1, while the condition 3 is the one where the dendritic cells were added in channel 3. If you compare to the one in condition 1 and condition 4, when no dendritic cells was added, what you can see is we have a decrease in activity. So more specifically here in MFA, in the channel where there is the co-culture between of neurons and dendritic cells. That's close it for this part. If you have any question, please, of course, ask at the end of the webinar, and I will let my colleague Helene talk about the PNS work.
Hi, everyone. So I will present first the work we've done regarding traumatic injury, and then the work on CIPN applications. So first, regarding traumatic injury. So we developed a repeatable and standardized axotomy protocol using the chip that is called the Dualink Delta. So the other motor or sensory neurons were added in channel 1 and let a load to grow toward channel 2, then channel 3. And we apply a solution with detergent, Triton, to dissociate the cell, create an injury on the axons only, not touching the cell bodies. So this is a picture showing you so either the motor neuron culture or the sensory axotomy. So here in the bright field, you will have the motor neuron before and after the axotomy. And it's here in the channel that you can, I'm not sure the bright field has the best one. But here you have, similarly, this is before axotomy on the motor neurons, and just 1 day after axotomy, when we can see that a few axons start regrowing and most of the axons were gone, that we really have cut all of them and not touch the other compartments. We tested different live staining, either with calcine or another red dye that will allow us to see the axon or the lack of axons by imaging, and better than in bright field. This is an example with the sensory neurons before and after the axotomy protocol. And we show that 100% of the neurons were absent 2 hours after the detergent exposure on both cell type. This is an example of how they regrow. So this is an example of the sensory neurons. 24 hours after the axotomy, you can see the growing in the channel 2, starting again, and 7 days axotomy on the delta. And this is on the right side, you have a blow up of this picture, this middle one, where you can really see all the projection, and you can see them in the microchannel and be able to see different axon or neurites growing. So the regrowth can be detected as early as 24 hours after, and 50% of the chip have axon reaching the channel 3 7 days only after the axotomy. We tested 2 drugs that are modulating the neurite outgrowth on both cell type. So nocodazole that destabilize microtubule, or the staurosporins that act on protein kinase and has a neurotrophic action. It's the same type of protocol, only the timing were adapted because of the different kinetic of the 2 cell type. So regarding the motor neurons, what we could see is that at a very high concentration of nocodazole, there is a cytotoxic effect. And this is the last graph on the bottom right that shows the vehicle and the high dose of nocodazole inducing cytotoxicity, but the lowest dose of noconazole 1 micromolar wasn't inducing cytotoxicity. So the effect that we are seeing of noconazole is only a reduction of the neurite outgrowth. When we tested the staurosporine, we could see an increase in the number of axons, an increase in the maximal length as well, and also the sum of lengths, showing you that you could, by using those mechanistic drugs or controls, you could build an assay in order to be able to test your compounds and know if you have a neurodegenerative or a neurotrophic effect. We did the same on the sensory neurons, and what we noticed first is that we had to reduce the concentration of noconazole, that 1 micromolar was preventing complete neurite outgrowth on the sensory neurons, but without affecting the viability. And we reduced tenfold the concentration, and we could see a small but not significant reduction of the noconazole. This inhibition of the neurite outgrowth was, at 1 micromolar, it was showing, in the number of axons, and the total sum of lengths. When we tested staurosporine, we used exactly the same concentration that on the motor neurons. We couldn't see the same effect of the staurosporine at the tested concentration. What is known about staurosporine, it has a dose-dependent effect. And what is the take-home message from this is that our platform can detect differences in efficacy of compound depending on the cell model. So the next step regarding traumatic injury and neurite outgrowth assays is to be able to look at the electrical activity and to encode axon regrowth, to be able to see the development of the new electrical activity as the axons are growing, the impact of neurotrophic compounds on this electrical activity, and can we have effects that are not wished, or kind of adverse effect from those neurotrophic compounds. And we could also confirm the functional recovery that neuron and axon is not only to project but to be physiologically active. And one of the advantages of the dual-link shift MEA is we have long microchannels that allow to follow the signal and potentially to see axonal activity propagation along those microchannels. Regarding our CIPN on-chip platform, so briefly, I will just remind you that peripheral neuropathies that are resulting from chemotherapy affect between 60 to 80% of patients during the course of treatment. That can cause dose reduction in some cases or treatment interruption, but mostly really a decrease in the quality of life because of this pain. And for 20 to 40% of patients, the pain will become chronic after treatment with really high impact on quality of life. There is 6 main categories of chemotherapy agents with really specific mode of action. And today we will focus on 2 of them, the platinum compounds such as oxaliplatin and the taxane compound with paclitaxel. And here you have on the right hand side, you have a description of those mode of action. Where are those compounds acting? So some of them, like paclitaxel, will act on microtubules, and nerve terminal will be where you have the effect. Whereas, for example, oxaliplatin will act as a DNA binder. The mode of action will be more through the on the soma of the cells. Current animal models in vivo sometimes can lack translationality. There is shows that some type of neuropathy are not detected in animal studies, and sometimes current in vitro assays don't always have the anatomical relevance or the complexity to understand certain mode of action or toxicity. So we started to develop the CIPN-on-chip platform using imaging. And we use the dual-link chips that are already being presented, and we grow sensory neurons from channel 1 to the channel 3. We waited until they are fully mature, and at day 20, we treat them with 2 compounds, so either oxaliplatin on the channel 1, where you have the soma, or paclitaxel on the channel 3, where you have the nerve ending. What we were able to show is for both compounds, but at different concentration, it's a dose response of degeneration. And we quantify this by looking at the surface covered by the axon or by the number of axonal debris in the channel 3. So for both, we quantify only the axon in channel 3. We just wanted the axonal impact and to try to reproduce this the dying back that is seen in CIPN. So the concentration were chosen to be consistent with the clinic and close to what can be found in plasma concentration from patients. And yeah, really for paclitaxel from 50 nanomolar, we had a dying back of neurodegeneration, and oxaliplatin, it was really clear at the highest dose of 100 micromolar. Then we decided to look a little bit further because the pain or the adverse effect that are experienced by patients during this treatment can either be during or can be more accumulative. So we tried to develop 2 types of recording or protocols, either to look after a long application of the compound, or to look at the direct answer of the compound. So we tested the oxaliplatin and we look at the thermal sensitivity of how the cell responding according to the temperature. So you have either the control recording, the recordings that were done before treatment that we name baseline, or showing you the recording after 72 hours. We can see that a change, so the axonal weighted MFR is increasing with temperature. And when we normalize by the baseline, so on the right-hand side, you have the normalization of the graph presented on the left. What we can see is that the response is different when we have the highest dose of 10 micromolar, and that the neurons are reacting more to low temperature than they should compared to the vehicle. And this is one of the symptoms of the pain felt by patients, that it's the sensitivity to cold temperatures. That so this indicates that we could use this model and potentially look if a preventive treatment could reduce those effects and then prevent further damage when those are accumulating. For the paclitaxel, so we tested the direct response of the application of the compound. So you have here electrode views of really of the axonal of axons. Yeah. So either in basal, so before the application of anything, when we apply the vehicle, we couldn't detect any high change in the activity. But when we added paclitaxel at 0.5 nanomolar, we saw a significant increase in the axonal activity. In this experiment, we treated sequentially, and the higher dose of paclitaxel at 5 nanomolar didn't increase the activity. We were expecting potentially more increase of activity, but we are guessing that we have a desensitization effect on the second exposure to paclitaxel. And we know the cell can respond because our positive control was clear when we use the KCR. So we can really detect acute effects that are not inducing cellular degeneration. Because from the previous slide, I show you at 10, when we tested 5 nanomolar, we didn't have any effect, morphological, phenotypic effect on the neurons. So this could be used, for example, to explore the capability or to test pain medications simultaneously, because chemotherapy are planned. Treatments could be developed to prevent those early effects and prevent future degeneration. What we're looking at is also to continue and to move closer to a full pain circuit by using the advantage of our compartmentalization by co-culturing sensory neurons with dorsal horn neurons. We are working on, starting to define the digital signature of the specific alter state, and also as a perspective, here we model CIPN, this one type of neuropathic pain, but other type of pain or neuropathic pain can be modeled in our systems. And this was the last slide, and I really thank you for your attention. I think now I'm going to let Thierry handle the questions.
Okay, thanks, Helene, and thanks, Benoît, for your valuable presentation. I think it was very interesting. We got some traction already from the audience, and we have some questions that are popping up. So my moderator is giving me back. So I'm looking at my screen at the same time. So there was first a general question thanking us for the clear presentation, and if some of those slides could be shared with the participants that could not join today. So and that could join, of course, as well. So we respond yes to both. So there would be some replay also of the webinar that you will get. That will be also available on our website. And additionally, we can send the PDF presentation to the relevant person that will ask for. So that's absolutely not a problem. So maybe I have some questions for you, Helene, talking more specifically about the CIPN application. There was one question about oxaliplatin, and so I'm reading the question: is oxaliplatin inducing acute hyperactivity like paclitaxel?
So our preliminary data that I didn't show here seems to indicate that it might have a different effect, and that the signature, both the two compounds are acting differently. We didn't see the same type of hyperactivity with oxaliplatin when tested, and more a really transient reduction in activity.
Okay. All right. Thanks for this precision. So in the same mood, and in the CIPN, it's more on the way and the benefit to use our organ-on-chip technology. So I think this could be addressed to you, Benoît. Can you describe the intended context of use for your organ-on-chip technology in studying CIPN? Is it primarily for drug screening, mechanistic studies, or both?
Well, I guess it can be used for both. I mean, it's a good question. I guess, everything should be starting with defining a context of use properly to make sure that, the question you want to answer. It's all going to depend on how you're going to, the kind of readouts you will have and how you'll be using it within the overall protocol. Both could be used. The first one, I guess, to try to screen for adverse effects. So have an idea, for example, if your new treatment is going to be generating neuropathies, or how likely it is to generate neuropathies in patients. And another one could be interesting to understand more actually how this neuropathy works and how you could maybe design strategies to help manage them and alleviate them.
Okay.
I guess.
Okay. Thanks for that. And I hope that the audience will get the feedback on that. So we have no questions that are coming, and there are many. So let me take them in not an order, I would say, but just as it comes. So which sensory neuron cell lines you use for CIPNs to die? So maybe I'll direct that to you, Helene.
So on this work, we use axonal sensory neurons. But we do have preliminary data indicating that other source can also model CIPN.
And I think the next question is could be a complementary one to the previous questions. Were sensory neurons from adult animals used?
No, we didn't use sensory neurons from adult animal. In this study.
Okay, fair. Another question about staining. Are all the stainings live staining? Question mark. Is it possible to stain the cells on MEA plate as in your slides? And last question around the staining. Is there a way to share the staining protocol? Maybe Jessica, you could answer that?
Sure. So yes, it's possible to stain the cells on MEA plate. Helene and Benoît show you some examples.
And this one example as well on the screen.
Exactly. And of course, it's possible to share the staining protocol. So just send us an email for that.
And just to add a little bit of precision, because of the first part of the question, so you can do some live staining, but you can also do just the stain, like the regular staining that you're using by fixing the cells and everything. Both can be done.
Okay, excellent. Thanks for this precision. Another question that is popping up. What specific endpoints or readouts do you prioritize in your organ-on-chip experiments to assess the development and progression of neuropathy or to model clinical readouts? So a question for you, maybe, Benoît, would you answer that?
Yeah, so there's a lot of different ones you could actually look at. So there are all the morphological one to make sure everything went well, of course. But the one we're trying to really push forward, I guess you would have understood now from the presentation, in nature are functional readouts. Because having cells that look good is a nice thing, but it's not necessarily enough. So that's why we actually rely a lot on the MEA to try to explore how they behave. And yeah, so I guess that's the one that is important. But once again, actually, just to maybe backtrack to the first question as well, it all depends on the context of use. So it's all going to depend on the question you are asking initially. We have a wide variety of readouts that are possible with this kind of technology. So don't limit yourself to the one you already know, explore new ones as well. And I guess that's the one.
I will add that you can combine with that. So have MEA readout and follow up your treatment culture. And then here you have an example of fixation and post-staining where you can check if you do beta-3 tubulin or cytoskeleton staining, look at your morphology, detect degeneration, but you can also combine with the supernatant analysis and ELISA, such as neurofilament, or do also some verify the cytotoxicity with LDH. So you can multiplex assays and really from one single culture.
Okay, excellent. So that's very comprehensive answer from both of you. Thanks, guys. Another question that about axotomy, and the question is, have you ever performed axotomy on other cell types, like rodent cells, for example? So Jessica, maybe you ready for that?
Yes. Thank you for this question. Yes, we have tested other cell types like primary, like rodent cells. such as cortical neurons and hippocampal neurons. What we see, it's possible to perform the same axonal injury using Triton, but we observe that the regrowth is not the same compared to the different cell type.
Okay, thanks for that, Jessica. I have other questions. I'm not sure they're in the right order, so I'm just trying to, put them together for the thematic. But let's go on. Let's deep into the Alzheimer for a minute. I have two questions. One is more related to the materials we're using. So, is there PDMS absorption of the oligomers? And the second question, still in the same range, is: have you tried in the same topic? Sorry, have you tried adding astrocyte in the co-culture? So, Jessica, maybe you are the right person for that.
So PDMS is very convenient to, it's biocompatible and also optically transparent, so it's possible to perform imaging, staining. But of course, it's possible to have some PDMS absorptions. We have a specific methodology to assess this point. Concerning the specific oligomers such as Ibita and Tau, we have adjusted the concentration on chip compared to devices.
And
concerning the
astrocytes,
we have not performed in this specific study, but we have a partner work on that.
And actually, once again, for those of you that will be lucky enough to get SFN at the beginning of next month, we have a poster actually on that, on the use of astrocytes. So, yeah, pop in and we can have a chat.
Okay, excellent. We have still a few questions left, so we have a few minutes. So maybe I can go and ask one more question. This time about CIPN. Is the model available with other cell types, motor neurons, donor cells, etc., and or cell suppliers? Helene, you want to answer that?
So yes, CIPN can be either sensory, neuropathies can be touching, sensory neurons and motor neurons. As we focus on the sensory neurons, and as I briefly mentioned, we did testing other providers. Other can respond to the stimulation and can be used for modeling CIPN. Here we only show you the data with our partner Axon.
Okay, excellent. In the same mood, I would say, comparing cells and this time on the traumatic injury, how can we explain the differences in results between sensory and motor neurons?
Very good questions. I cannot explain it. I can say that the platform can detect differences. We didn't go into the mechanistic details of why those compounds are acting differently. I'm guessing that the sensory neurons are more kind of fragile or affected by the drug like the nocodazole, and also because of the different of kinetic of growing, it is most likely also that those drugs are affecting them differently.
Okay, excellent. So I think that we are going to the time. So we are ready to answer other questions also apart from the webinar session now. So feel free to ask us, any request that you have on that, and remember that there is a replay also at the end. So thank you very much for participating to this webinar, and we wish you a good day or a good evening. Thank you.